pleasure and an honor to give one of these Lucy Cherbas's lectures. I didn't know Lucy Cherbas personally, but I certainly know of her legacy, her commitment to generating community resources, generating tools that can be broadly shared within the community. And I think that spirit has been responsible largely for the great success of Drosophila in general. And I think if you think about it, many organisms have served as models for studying major biological questions over the years. But there are very few that have had the same long run of successes that Drosophila has had. And I think it's due to the fact that there are people like Lucy Cherbas who've had a commitment to not only generating tools, but making them broadly available to the community that has fueled the success of work in flies. And I think the other component that has been important is that those of us who work on Drosophila have not just been interested in developing tools, but also in using those tools to reinvent Drosophila as a preparation to keep it relevant to critical questions in biology and use Drosophila to study those questions. And so those have been very much advances that have been fueled by the generation of new tools. And I just wanted to sort of start by trying to put that in some historical context here, where I've divided the history of research in Drosophila into what I see as sort of three major eras. And the first, of course, was this sort of foundational era in genetics that relied on the discovery of spontaneous mutants, such as the discovery of the white mutant, that influential sort of first discovery that T.H. Morgan made, and the discovery of recombination that allowed genes essentially to be realized as inheritable or heritable elements aligned along chromosomes. And then Muller's recognition that ionizing radiation could be used to induce mutations and, in fact, induce changes in chromosomal structure, and the exploitation of polytene chromosomes of salivary glands to visualize chromosomal rearrangements and align the relative positions of genes along chromosomes. All of this was part of the sort of classical period that really defined the beginning of genetics, and where Drosophila played a really critical role. And that role was recognized by a couple of Nobel Prizes, one to Morgan, one to Muller. In this period, I think sort of gave way to a second period in which Drosophila played a major role in elucidating the genetic pathways underlying complex biological phenotypes, those involved in, for example, behavior or development. And these were organismal phenotypes where the methods of molecular biology that had been developed in the 50s and 60s didn't really apply, because the simple organisms that had been used then, phage and bacteria, didn't have behavior and development in the way that most metazoans do. So Drosophila again came to the fore. And this was actually, again, due to the technical development, the development of chemical mutagens like EMS that could be used to introduce point mutations at scale, where you could really do saturating mutagenesis screens to identify genes involved in these generating these complex phenotypes. And again, this work was recognized by a couple of Nobel Prizes indicated here. And I think for me, this particular era in the fly is best exemplified by a conversation that I had with a colleague when I first started my lab at NIH. And I told him I was working on Drosophila, and his response was, "Oh, well, what gene are you working on?" And I wasn't working on a gene. I told him I was interested in behavior, but that in fact, since behavior was not in general a direct output of genes, but rather was determined by cells and the unique signaling used by the nervous system, that we were really more interested in cells and circuits. But that those processes could also be addressed using Drosophila. And that's due to, I think, what I'm calling here sort of the third era of Drosophila research, which was really heralded by this seminal development of a technique for transforming flies, P-element mediated mutagen or transformation, introduced by Jerry Rubin and Alan Spradling in 1982. And this, by allowing foreign transgenes now to be expressed in the fly, it meant that you no longer had to rely on manipulating native genes in order to see their effects and phenotypes, but you could actually introduce foreign genes that could be used to study and manipulate the activities of cells. And that whole methodology was really underwritten by the development of this fantastic GAL4 UAS system that was developed by Andrea Brand and Norbert Perimon, which allowed you to, again, go in and express genes in a targeted fashion in particular cell types and manipulate the function of those cells to study their activity, their contribution to phenotypes. And I think this, this methodology, this approach has been nowhere more important than in the study of the brain. And neuroscience in the fly, of course, has really come to the fore over the last couple of decades. And as one has seen, it progressed with the advance of particular techniques. And those techniques some of those techniques are ones that my lab has helped develop and I'll be talking about today. But again, I wanted to just stress that neuroscience, which was what I was trained in, as Daniel mentioned, I actually came into Drosophila fairly late in my career, late in my postdoctoral career. Worked on other model organisms, but I was keenly aware of the lack of certain methods in neuroscience that where the fly might be useful. And so one of these was articulated in Francis Crick's scientific autobiography, With Mad Pursuit. And I've been interested to see over the years how many people were similarly influenced by this particular quote that Crick made, as I was. So he, in talking about sort of the crudeness of the methods available for ablating or destroying parts of the brain in order to see what they did, Crick said, for example, it would be useful to be able to inactivate, preferably reversibly, a single type of neuron in a single area of the brain. And so I think what galvanized me upon reading that particular quote was that I thought I saw a way that one might be able to do that. As Daniel mentioned, my background was in ion channels, and I was working at the time in Len Kazmarek's lab at Yale, where there was a real focus on potassium channels. And potassium channels are really sort of the brakes of nervous system activity, and they're targets of neuromodulation, which was also a strong interest of mine. And the biggest game in town at that time, this was the mid-1990s, was the Shaker channel from Drosophila, which had been cloned about a decade before and had been intensively studied in sort of structure function analyses to determine which parts of the channel did important things. And my realization was that one might be able to use that knowledge to re-engineer the Shaker channel into something that might make it a great tool for silencing neurons. And the idea was to introduce certain point mutations and other changes that would allow this channel to activate at much lower voltages than it normally activated, so that it would activate roughly with sodium channels and short circuit a brain cell's attempt to become active. And I was working on a plesia at the time, again, as Daniel mentioned, was able to show that this re-engineered shaker channel that I developed was able to effectively silence neurons from that animal. So in response to a current injection where you'd normally see action potentials generated, cells expressing this modified shaker channel were effectively silenced. And so in a fit of hubris, sort of in retrospect, I named it the electrical knockout channel for suppressing excitability. And my goal here actually was not to take this into Drosophila, which would have been a natural choice, but was more interested in using it in the mouse. And as I say, my interest was in neuromodulation and behavior. And the hypothalamus in mammals is sort of a critical node in the brain for generating instinctive behaviors and highly reliant on the use of neuromodulatory factors to affect that. And so I wanted to take this channel, the eco channel, and direct its expression to different cell types in the hypothalamus. And the problem was that after about a year and a half of failures in getting my eco channel to express in mouse neurons, it seemed to get hung up in some intracellular compartment and never be able to do its job of silencing neurons. It occurred to me that since this was actually a Drosophila channel, it might actually perform better in flies. And so at this point, I took my tools to Haig Kashishian at Yale, whose lab I knew was also interested in issues of excitability. And I was actually ready just to turn over my tools to Haig because I was actually thinking about dropping out of research after numerous failures. And Haig, I think, in sort of characteristic Drosophila fashion, consistent with the culture, said, no, you need to come to the lab and try this wonderful idea. Let's try it out and see if it works. And so Haig set me up to work with Thomas Osterwalder, another postdoc in his lab. And I was impressed that within short order, we were able to make fly lines that expressed UAS, ECO, and were able to use these enhancer trap lines that had become increasingly available over the last few years to drive its expression in different parts of the nervous system. And so one of the first tests that we put it to was to use a fly line called A307, which was known to express in the giant fiber neurons responsible for the jump reflex in Drosophila, and wanted to see if I could suppress its behavior. And much to my surprise, I actually got a phenotype that not only effectively reduced the ability of the flies to jump, but had much more profound effect on developmental process, namely the expansion of the wings. And at that time, I knew very little about flies, nothing about wing expansion. But the more I read about it, the more interesting it became to me, because it turned out that wing expansion was actually the last step in this behavioral sequence, the so-called adult ecdysis sequence, that basically got the animal out of the pupal case and then allowed it to complete metamorphosis by expanding its wings in order to make them flight worthy. And this process had been studied for several decades in Jim Truman's lab, later by Mike Adams' lab, and had been found to be driven by the cascade of hormones and neuromodulators, including ecdysis triggering hormone and eclosion hormone, and then downstream of those, another pair of hormones that were co-expressed typically in the same neurons, CCAP, or crustacean cardioactive peptide, and bursicon. And so this type of neuromodulatory action that was responsible for generating an innate behavior seemed very similar to me to the types of behaviors and mechanisms that I had been interested in exploring in mice, and seemed like a great opportunity to do something similar in flies. And so I quickly gathered all the enhancer trap lines I could find around in Haig's lab and around Yale generally in fly labs, and conducted a screen using the enhancer trap lines to drive eco expression typically 2 copies of ECO, which seemed to be sort of the sweet spot for getting phenotypes without causing lethality, and was able to identify 24 of those lines that in fact gave varying degrees of flies with deficits in wing expansion. So where the solid shading here indicates the fraction of flies that could not expand at all, and the gray, the partially expanded wings, and so on. So there were 24 lines, and these were the lines that I took to my lab when I started at NIMH. And the goal here was to identify the neurons within the expression patterns of these different lines that were responsible for the wing expansion deficits. And the goal was that, or the hope was that each of these would identify a different subset of the brain cells that were responsible for wing expansion, and that by piecing together those different cells, it would be able to construct or reconstruct the circuit that was responsible for wing expansion. So as you'll see, those hopes were quickly dashed. When I got to NIMH, a paper appeared shortly thereafter from John Ewer and Jay Park's lab, in which they showed that neurons that expressed crustacean cardioactive peptides, so CCAP, could lead to wing expansion deficits. So they made a CCAP GAL4 line. They showed that if you ablated those neurons, flies didn't expand their wings. And so I requested that line. We used it and showed that if you drove 2 copies of ECO, indeed, these flies could not expand their wings, indicating that the neurons that express CCAP were at least one critical node in this circuit. And Haozhan Luan in the lab, early postdoc, who was spearheading this project, also quickly made a GAL80 line under the control of the CCAP promoter, so that we could express GAL80 in the CCAP neurons and determine which of these enhancer trap lines might include those neurons, and that they might therefore be functionally responsible for the generation of the unexpanded wing phenotype. Well, much to my surprise and chagrin, almost all of the lines showed a very strong reduction in phenotype when expressing GAL80. And that indicated that, in fact, the CCAP neurons were a component of these enhancer trap lines, and they were, in fact, the ones that were functionally important for generating the unexpanded wing phenotype. So the idea of reconstructing the entire circuit disappeared at this point. But the good news was that we had at least one critical node in the circuit, the neurons that express CCAP. And the other thing that we were able to do in relatively short order is show that this subset of CCAP neurons that expressed this other critical hormone and neuromodulator, bursicon, were in fact the ones that were functionally important. So if we targeted only the subset of CCAP neurons that express bursicon using a bursicon GAL4 driver, we were able to also generate a significant wing expansion deficits. And so this told us that the bursicon expressing subset, in fact, was critical for wing expansion. So at the same time, we were also very interested, though, in having a test that would allow us to determine not just which cells were necessary, but also which ones, when activated, might be sufficient for driving a particular behavior. And so in our case, for inducing a wing expansion in flies that might not otherwise perform that function. And so during this period where I was starting my lab, we were also actively looking for tools that were appearing in the literature, particularly ion channels, that might be used to increase the excitability of neurons so that we could stimulate them using, again, in a targeted fashion, using the GAL4 UAS system. So one of the tools that or one of the channels that came to our attention was this bacterial sodium channel called NATCHBAC, which would significantly increase the excitability in cells. And we made correspondingly made UAS Natchback lines that in which Natchback was also GFP tagged so that we could visualize it. And we showed that if you express Natchback in the eye and subjected the eye to a light pulse, that the electroretinogram now had this enormous spike indicating that the excitability of those cells had been increased substantially by the expression of Natchback. So this was a useful tool. It turned out that as we were developing this, that Mike Niederbach and Todd Holmes were working along the same lines and had also made Natchback lines for use in Drosophila. We ended up exchanging reagents and publishing jointly on this. Another ion channel of interest, and one that in some ways was more interesting and that would allow temporal control, was the cold and menthol receptor from rats. And so this is a protein encoded by the TRPM8 gene and expressed in rat sensory neurons, where it senses decrements in temperature. And one can see from this graph here that the channel, in fact, becomes increasingly active. So this is increasing current in this axis with temperatures that range over the temperatures that are useful actually for growing and analyzing Drosophila. And we similarly made UAS TRPM8 fly lines and used them to express in motor neurons, and showed that, in fact, with decrements of temperature, we could elicit spiking activity in motor neurons that could be recorded in the muscles, and that depending on how many copies of the TRPM8 gene you expressed, you could get more or less excitability. So this was a very encouraging. I should say that we also took a long look at this light-sensitive channel called channel rhodopsin, which was published in 2003, and fatefully decided that light probably wasn't the best stimulus to be relying on, because it was a potent behavioral stimulus in its own right in flies, and also the problem of getting it through the membrane into deeper tissues seemed formidable. So, well, we didn't make UAS channel rhodopsin lines, and the rest is history, but only have a few minor regrets about that. So TRPM8, though, was an important and useful tool to us in the beginning, and the utility is shown in this particular video, where you see three different flies, one of which expresses TRPM8 in the CCAP pattern, the other in the more restricted pattern of cells that express only bursicon, and then a control fly. These were subjected to a 15-minute temperature shift to 18 degrees to activate TRPM8, and then return to 25 degrees. And you can see that the two flies that were subjected to the temperature shift and that were expressing TRPM8, in fact, respond by expanding their wings rapidly in this confined environment that is normally inhibitory for wing expansion, and as you can see from the control flies. So this type of experiment told us that the bursicon expressing neurons were in fact not only necessary for wing expansion under normal circumstances, but could be used to drive wing expansion. They were therefore sufficient for inducing that process when activated. Oops. So what are the bursicon neurons? Well, they come in sort of two anatomical flavors. There's a single pair located in the subesophageal zone, and these have projections that go throughout the nervous system, up into the brain and then down into the ventral nerve cord. And then the other set is a group of 14 located in the abdominal ganglia. And these have a much simpler projection pattern. So this is an example of a single one made by flip-out methods, where you can see the axons projects out of the nervous system and then goes to contact targets in the body wall. And these guys express substantial amounts of bursicon, as can be seen from immunostaining in their axons, and that's simply released into the hemolymph. And so we had a strong hypothesis that these neurons in the subesophageal zone, which we called the BSEG, were probably responsible for inducing behavior, probably necessary for behavior. Whereas these were likely to play a role that was different from actually performing behavior. And in fact, it was known that bursicon was required for plasticizing the wings, making it possible for them to expand. And so these seemed likely to play that role. So what we needed was a tool for selectively targeting these different populations of bursicon neurons so that we could selectively activate or inactivate them. And that meant a tool that provided more refined expression than what we could achieve at the time with GAL4 UAS. And so Hao Zhang, again, who was spearheading this work, settled on a strategy which we call the split GAL4 system. And this was based on the technology underlying yeast 2 hybrid system, for any of those who may remember it. And the yeast 2 hybrid system capitalized on the modularity of the GAL4 transcription factor, the fact that it could be broken into separate domains, a DNA binding domain, or DBD, and an activation domain that activated transcription. And that you could, by rejoining those fragments, reconstitute transcriptional activity. And so what Hao Zhang did was couple each of these 2 fragments to a pair of heterodimerizing leucine zippers, so that whenever they were co-expressed, they would come together to reconstitute activity. And the benefit of splitting them, of course, was that then each component could be separately driven by a different enhancer to a different set of cells. And only those cells that express both components then would be transcriptionally competent and be able to activate transgene expression at the intersection of those 2 patterns. And so using this system, Hao Zhang was able then to subdivide the bursicon population of neurons into these 2 component populations. He made a GAL4 DBD that was expressed under the promoter for bursicon in general, and then made enhancer trap lines that expressed the VP16AD activation domain in different patterns. And so by combining these 2, he was able to isolate 2 different lines. I should mention that one issue that arose in this work was that the GAL4 activation domain itself proved to be very weak in this particular configuration. And so we used instead a different viral transcription activation domain, VP16AD, which gave very robust expression. So with these tools in hand, then we were able to do the same type of activation experiment that I showed you earlier. In this case, we have a fly that is expressing the TRPM8 channel only in these 2 neurons, the BSEG, one that's expressing the TRPM8 channel only in this subpopulation of bursicon neurons, and then appropriate controls here. These, again, were subjected to cold temperatures for 15 minutes, returned to 25 degrees. And you can see that only the animal that is expressing TRPM8 selectively in the BSEG neurons has stopped and expanded its wings, again, in this inhibitory environment. And the flies in which this component of the bursicon population was activated did not respond with wing expansion. So this allowed us to determine that, in fact, this single pair of cells in the subesophageal zone, the BSEG, were sufficient for driving wing expansion. So another natural question, as I said, we were interested in understanding sort of the broader circuit involved in wing expansion. And one key question was, well, if bursicon is likely to be a key signaling molecule, who's it signaling to? Who's downstream of bursicon? And so what we wanted to do was target cells that express the bursicon receptor, which was encoded by a gene called rickets. And the sort of principal methodology for doing this kind of targeting at the time was homologous recombination, in which one inserted the GAL4 coding region at the site of the ATG start codon for the gene of interest. And so Feng-Chu Diao in the lab, who is driving this project, did this with great labor, used homologous recombination to insert GAL4 at what we thought was the start codon. So I should say that the problem here was that although rickets had been identified as the receptor for bursicon, the actual start site was not identified, and there were several candidates, one of which, however, looked much better than the other two. So Feng-Chu inserted GAL4 at that site, and he got beautiful mutants that failed to expand their wings, but he got no GAL4 expression. And so clearly we had mistargeted GAL4. And it was about this time, though, that 2A peptides were sort of coming into consciousness. Certainly, I'd learned about them at about this time. And 2A peptides, for those of you who don't know them, are viral peptides that are used by viruses typically to allow the translation of multiple products from a single transcript. And they do that by virtue of a consensus sequence at their C terminus that has a particular motif that promotes ribosomal skipping. So what this means is that a ribosome that's translating a particular messenger RNA will translate up to the point where it reaches this glycine proline motif at the end, at which point it will drop the translation of the product that it has been translating, and then renew translation on the same messenger RNA from the standpoint starting with the proline. And so this, again, is a method for generating two different proteins from the same messenger RNA. And what we realized was that if we incorporated a T2A GAL4 module into a gene of interest, into, say, a highly conserved site that we knew was going to be translated, we would be able to generate a truncated translation product of the protein of interest connected to this 2A peptide, but then independently also the GAL4 translation product that could go and do its business. So this is a way of coupling GAL4 expression to the expression of a gene of interest. And Feng-Chu used this method to, again, use homologous recombination to insert T2A GAL4 into this very conserved hinge region of the rickets protein that was in front of the transmembrane region. And by this means, couple GAL4, as I say, to the expression of rickets. And we call this method the T2A GAL4 inframe fusion technique. And found that, in fact, it worked very well. Feng-Chu was able to generate lines that allowed us to visualize the pattern of expression of rickets. It was distinct from the pattern of expression of bursicon. So these were the neurons that were downstream of these cells. We were surprised to see how many there were, but at least they were there. And we were able to convince ourselves that, in fact, these were the relevant neurons, because when Feng-Chu took the driver line for the bursicon receptor and used it to drive a rescue construct for rickets or the bursicon receptor, those flies that expressed the rescue construct under the control of the driver expanded their wings. So clearly we were in the right ballpark. It was in the course of writing up that paper that we realized that, in fact, this T2A GAL4 method could be more broadly applied. And in fact, that one application would be to combine it with the recently published mimic technology that came out of Hugo Bellin's lab as part of the Gene Disruption Project. And so the mimic insertions developed by the Bellin lab were placed between these ATTP sites that were recognition sites for 5C31 recombinase, so that they were modular. And they had a GFP gene that was placed between a splice acceptor and a donor site, so that when the mimic insertion was in an intron between two coding exons, this region would get incorporated into the transcript along with the native, or for the native protein, and that this gene inserted within the mimic construct could then be translated together with the encoded protein. And so what Feng Chu did was develop constructs that again had these ATTP recognition sites and a splice acceptor domain with a 2A peptide, so that we could either use them to replace an existing mimic insertion or place them de novo into an intron of interest using CRISPR-Cas. And that what would happen was that this 2A gal4 would be incorporated into the native transcript, and then during translation, the gal4 would be independently translated. So again, a way of coupling gal4 expression to the expression of a native gene of interest. And Feng Chu made lines that could be useful not only because they express gal4, but also made constructs that allowed for the modular replacement of any gal4 or mimic line or construct with any of a number of other transcriptional factors that might be useful, including gal80, the transcription factor LEX-A, and then of course split gal4 components. So this was a way of really sort of facilitating the gaining genetic access to cells that specifically expressed a gene that one was interested in. So I don't have time to take you through Feng Chu's subsequent work, but I just wanted to say that was by combining this Trojan. So I think I forgot to mention, we called these Trojan exons. And that was by analogy to the Trojan horse, which according to Greek mythology, was used to ferry soldiers into the town of Troy. And then the soldiers emerged from the horse and took charge of the city. And so this seemed like an apt analogy, because essentially the technique was a way of introducing a foreign exon into a gene of interest and then allowing it to release gal4 to essentially take control of whatever aspects of cellular function one wanted to. So we called it the Trojan exon method. These Feng Chu used the Trojan exon method together with split gal4 in order to subdivide the neurons that express the bursicon receptor into components that use acetylcholine as a neurotransmitter, and another subset that use glutamate as its transmitter. And he showed that, in fact, these glutamatergic neurons were motor neurons that were responsible for driving some of the key critical behaviors underlying wing expansion. And the cholinergic subset were neurons that in fact participated in a positive feedback loop with the source of bursicon, BSEG. And this positive feedback loop essentially allows the system to adjust the temporal control of release of bursicon and coordinate it with environmental input, particularly that inhibitory confinement that I had mentioned and that you saw in those previous videos. So we have an increasingly sophisticated model for the circuitry underlying wing expansion. But also of great interest to us was, what about the circuitry that governs this initial step in the ecdysis, the adult ecdysis sequence, namely eclosion? How does the animal actually succeed in getting out of the pupal case so that it can expand its wings? And this is there was very little knowledge about the behavior that flies perform prior to eclosion, principally because it's very hard to see what an animal is doing when it's inside the pupal case. And so here we took advantage of a technique. Oh yeah, here we go. Took advantage of a technique that we had exploited previously when studying pupil ecdysis, and that was to make a fly line that could drive expression of transgenes throughout the musculature. And so what we needed specifically was a tool that would not only drive expression of a transcription factor, but we wanted to be able to drive drive LEX-A so that we could use this LEX-A to visualize what was happening in the musculature, while at the same time we used GAL4 lines to manipulate neuronal activity and saw how it affected the muscular activity. And so again, this is sort of a tribute to the sharing of resources in the Drosophila community. It went back to my original screen that I'd done as a postdoc. There was one line that actually was resistant to the effects of GAL80, clearly gave unexpanded wings due to some component that was not the CCAP neurons. And in fact, when I had analyzed these lines, I considered them not particularly relevant because this driver expressed throughout the musculature. So it was a pan-muscle driver and simply inhibited expansion because it was inhibiting muscle activity. And this was one of the NP lines, NP3137, and it came to the fore as a line of interest when we were looking for a pan-muscle driver that we might be able to make a LEX-A driver from. And using the Japanese database for these lines, we were able to find that, in fact, this insertion was located just 5' of a gene that was known only for its lethal phenotype, and that gene, in fact, had a mimic insertion in a coding intron. And so within a matter of a few short months, we were able to make lines where we replaced the mimic insertion with the Trojan GAL4 and substituted the Trojan GAL4 with the LEX-A to make a line that had expression in the LEX-A pattern. And so this allowed us to then visualize the activity in animals prior to eclosion. What you're seeing here is GCAMP expressed throughout the muscles. This is activity prior to the release of ETH. And what you see is that a pattern emerges of high frequency activity in A1 muscles of the first abdominal segment. It dies out after a while and is replaced by this other very dramatic pattern in which broad activity of muscles induced in what we call eclosion bouts, and those eventually lead to eclosion of the animal. So this gave us a handle on at least two key patterns of activity that were associated with eclosion in the fly. And I need to take a step back. A key observation that had been made by the Truman Lab was that although ETH and EH were critical for driving the process of eclosion, these two seemed to act as positive regulators of that event, and that in fact there was evidence also for pathways that negatively regulated eclosion. And Mike Adams' lab had proposed that, in fact, those neurons might be targets of ETH, but a particular subcategory of those neurons that express the B subtype of the ETH receptor. And so another sort of experiment that we were able to do was to inhibit activity in specifically these neurons that express the ETHRB, and showed that in fact we could inhibit eclosion. So when we used a different temperature-sensitive ion channel, TRPA1, introduced by Paul Garrity's lab, very potent inhibitor, into these neurons, we showed that as long as we were activating these neurons, the flies could not eclose. As soon as activation was relieved, they would eclose. So this indicated that there was at least a negative pathway regulating eclosion that used these ETHRB receptor neurons. And so we were interested in using this technique for monitoring muscle activity to discover what actually the ETHRB neurons were inhibiting. So going back to the video that I showed you there were two major patterns of activity in the musculature, one of which involved the rapid activity of these A1 DIOM muscles. And we were able to show, using other markers for behavior in the fly and key events, that this in fact correlated with the resorption of molting fluid. So molting fluid is secreted by the epidermis prior to a molt in order to dissolve components of the exoskeleton, which has to be shed. And the A1 activity correlated with the resorption of that fluid. And it occurred roughly 5 to 10 minutes after the release of ETH. Now, the second major pattern of activity, those eclosion bouts that I showed you, actually followed the release of EH, and they were responsible for inflation of the talinum, which breaks open the operculum. And by monitoring the muscles of these two kinds, we could isolate that activity. Shown here in orange are the resorption bouts of activity by monitoring DIOM activity, and then by monitoring muscles in the head, we could visualize these eclosion bouts. And so the question we wanted to ask was, what happens if we stimulate the ETHRB neurons, which we knew blocked eclosion? What happened? And this shows the results of an experiment where ETHRB neurons were activated after the onset of the resorption bouts. And what you see is that the animal simply continues to execute resorption bouts, and that eclosion bouts are inhibited. So the simplest explanation of this, which proved to be true, was that there are at least two components of the ETHRB population, one of which promotes the resorption bouts and one of which suppresses the eclosion bouts. And so, again, don't have time to take you through all the data, but what Haozhang Luan, again, spearheading this project, was able to show that, in fact, it's a cholinergic subset of the ETHRB neurons that is responsible for generating this A1 neuron activity, the resorption bouts, and that it's a set of monoaminergic neurons that express the vesicular monoamine transporter VMAT that are responsible for suppressing the eclosion bouts. Now, the surprise was our initial guess as to what was suppressing the eclosion bouts was that it would be neurons that used one of the main inhibitory neurotransmitters in the fly, either GABA or glutamate. And when we had shown that if you stimulate the glutamate expressing subset of ETHRB neurons, that you could inhibit eclosion just as efficiently as with ETHRB itself. But when we activated these neurons and looked what happened behaviorally, we found that actually you got a promotion of eclosion bouts, but that eclosion itself was still blocked. And so what this has led us to is a model of this type, where there are actually at least three different components or three different pathways that involve ETHRB expressing neurons. One cholinergic that promotes resorption bouts, and two that are inhibitory, one that uses monoamine transmitters, the other of which uses glutamate, and these block two additional steps in eclosion. So eclosion turns out to be a somewhat more complicated process than we'd originally imagined, involving multiple steps, and our current efforts are to try to understand how eclosion hormone itself is integrated into these pathways. So I'm reaching the end of my time here. I just wanted to conclude by talking about a couple of the other techniques that we have developed recently that used, as Daniel mentioned, the split intine technology. Split intines are naturally occurring protein fragments that occur at the N and C termini of major proteins that themselves are split. And what happens is that when these two proteins, which are typically controlled independently by different promoters, when they're expressed in the same cells, the split inteins come together and self-splice so that they remove themselves and religate or ligate these other two fragments into a full-length protein. This, again, is a strategy used by certain microorganisms to generate proteins from two precursor molecules. Again, I don't have time to describe it, but Haozhang Luan had gotten very intrigued by this, exploiting this particular mechanism, and designed a very elegant system, which he called Sparklin, which used a triply split free molecule in order to execute a stepwise restriction of expression of a GAL4 line, using, again, sort of involving steps that I don't have time to go into here. But two other methods that we have generated that I think are likely to be broadly useful to the community, and that we have contributed to as part of a collaboration with Norbert Paramon's lab, are a split GAL4 system that uses split inteins and is inhibitable by GAL80, unlike the original split GAL4 system. And so here, this was a technique that Haozhang, again, had already been sort of experimenting with when the Paramon lab contacted us and asked if there wasn't possibly a way to perform a split, or to make a split GAL4 system that was responsive to GAL80. And so Haozhang used split inteins to separate, again, major portion of the GAL4 DNA binding domain from the rest of the GAL4 molecule, so that when these came together in the same cells, they could drive transgene expression, and that expression was now suppressible by GAL80 because we were reconstituting the GAL80 molecule itself. And so this work has really been brought to fruition by Ben Yu and Campen in the Paramon lab. And this is just a figure from this recent paper showing that if you use this split intein GAL4 and to drive expression, that at the restrictive temperature of GAL80, you see inhibition of the GAL4 activity. But if you move to the permissive temperature of 29, GAL4 activity is revealed. So that system is useful. At the same time, we also worked with the Paramon lab to convert another system, the gene switch system, which has inherent temporal control, to a split intein based system, using, in fact, exactly the same split site as used in GAL4. So gene switch, for those of you who don't know it, has a GAL4 DNA binding domain coupled to a P65AD activation domain. They are separated by the ligand binding domain for the progesterone receptor, which binds a small molecule RU486, which allows these to become transcriptionally competent. So again, Haozhang was able to demonstrate, again, with Ben Yu and Campen's help, that these components could be separately expressed, and then in cells that express them, they would reconstitute gene switch activity, but that it was completely reliant on RU486. And again, this is a figure from Ben's paper showing that in the absence of RU486, you don't get expression of reported GFP, but you do in its expression, again, using this split intein gene switch. So these are split systems that now offer the possibility of not only restricted expression, but temporal control. So lastly, Feng-Chu Diao in the lab has also used the split intein technology to make a more refined Trojan exon method. One of the issues with the original Trojan exon method, as I mentioned, is that it generates a truncated version of the endogenous protein that can sometimes be deleterious if that fragment, that truncated fragment has dominant negative effects. And so Feng Chu used the split intine method to design a system which would actually religate the two halves of the native protein into a full-length protein while also generating independently a Gal4 molecule. And as a proof of concept of this, he made constructs that expressed this split intine Trojan in all of the different patterns of the Hox genes, and made constructs and lines that were incorporated split Gal4 technology. So this shows the glutamatergic subset of neurons in each of the different Hox gene patterns of the nervous system. And if you take any of these patterns, you can further use split Gal4 then to subdivide according to, for example, neurotransmitter type or any other cell feature of interest. So this shows the cholinergic, glutamatergic, GABAergic, and monoaminergic subsets of the sex cones reduced neurons in the nervous system. So again, I think this is likely to be a broadly useful tool to the community. So just to come back to community, I wanted to say that we have benefited enormously in our efforts from the efforts of other people within the community. So each of our major tools, split Gal4 and the Trojan exons, were in fact made more broadly useful by the efforts of others. So for example, Jerry Rubin was an early adopter of the split Gal4 method and introduced a certain number of changes to it and applied it in a sort of high throughput manner to make thousands of split Gal4 lines together also with Barry Dixon's lab. And these have all been deposited at the Bloomington Stock Center for use by the community. Trojan exon methodology was taken up by Hugo Bellin. Hugo Bellin's lab as part of the Gene Disruption Project. They made, again, thousands of lines that incorporate that technology. Again, some of it in a slightly modified version of their Primic construct. But again, those lines are all broadly available to the community through the Drosophila Stock Center at Bloomington. And then all of our constructs we've deposited with the DGRC. Again, thank you to the DGRC for your work to make tools accessible to the rest of the community. And then I just wanted to close also by saying that one of the resources that all of us rely on, certainly my lab relies on every day, is Flybase, which currently needs the help of everybody in the community because of complications related to funding of that. So I hope we can all join together to rescue that very important resource to all of us. So with that, let me close. Just wanted to thank you as my audience. I've gone a little bit over, but also my lab that has participated in the work that you have seen, and to my many collaborators who have helped generate the tools that I've talked about today. So thank you very much.
All right. Thank you, Dr. White. And for Q&A, like I said, send a message out. If you have questions, you can put it on the chat. You can also just raise your hands and ask directly. We'll call on you to do that. And we'll just give people some time to process. But meanwhile, I have one question. You talked about the resorption phase. Is resorption activity, is it controlled by, is it neuronal control as well?
Yes. So resorption resorption happens slowly over several hours prior to eclosion. But the phase that I was showing there, or was talking about there, is a phase that is active resorption that requires the activation of the pharyngeal pump. So the animal is actually swallowing the remaining molting fluid using its proboscis. And so we've also shown, I didn't show this, if you look specifically at the muscles of the pharynx, you can see that those also become highly active at the time of the resorption phase. So the activity of the A1 muscles, we're actually not sure what it's responsible for behaviorally but it is a correlate of that resorption phase activity. It happens coincidentally with the activation of the muscles of the cervarial pump. And that pumping activity is under neuronal control.
Okay, thank you. That clarifies a lot. I thought it was a passive kind of thing at first.
Yeah, well, so some resorption actually probably does take place passively through the epidermis. But as I say, as the fly approaches eclosion, there's this very active phase where the rest of the molting fluid is very quickly taken up. And that seems to be, as I mentioned, in response to ETH secretion.
Thank you. Tom Kaufman left a note saying it's a nice talk.
I'm glad he liked it. I hope he liked the Hox gene patterns and tools.
Okay, any questions from anyone? Feel free to put it in.
Hope it didn't all go by too quickly.
I think we are just on time. Perfect. Yes. Mhm.
Okay.
Yeah. We'll give everyone like a good 15 seconds, and then we'll just thank Dr. White again if there are no questions. Okay, so what's next? Okay, Kale has a question. Kale Whitworth from BDSC, he asked, what is next? What's the next set of tools that you are going to make?
So that is a great question. So the tool that I would really like to have at this point is an alternative to GCAM. So GCAMP is a great tool for monitoring neuronal activity, but it requires, because it's fluorescence based, it requires illuminating the fly constantly with blue light or light of another wavelength in order to see the readout, to be able to detect calcium activity. And there are a couple of problems with that. One is that you need the light to actually penetrate to the neurons that are of interest, and you need the light that is emitted by GCAMP to then move back out through the intervening tissue to the microscope lens so they can visualize it. And those are not problems that are necessarily insurmountable, but you sort of need the right type of neurons in order to be able to monitor. And it's difficult, if not impossible, to do in an animal that is moving around constantly. And so the tool that I would love to see is a bioluminescent calcium indicator. So one that uses luciferase instead of GFP as sort of its primary component, and that doesn't require an illuminating light in order to see activity, but instead just emits light in response to endogenous calcium elevation. And the beauty of that would be that you could have it as long as you could target specifically the tool to a particular set of neurons, you could monitor the activity of those neurons while the animal was in the dark, freely behaving, and then correlate that activity with the movements of the animal. Again, it would require sort of periodically monitoring neuronal activity and periodically monitoring behavioral activity, but it would be a way that you could monitor activity in a freely moving animal without having to constantly illuminate it and depend on the light signals being retrieved. So that's a tool that, spoiler alert, we are working on. But if anybody else is working along those lines and is making progress, we'd love to talk to you. As I say, many of our tools have been developed in collaboration with others. We welcome collaboration, and so if you're doing something along those lines, love to hear about it.
Well, thank you. I think that's all. Yes, so let's thank Dr. White one more time. And the recording for this seminar will be up, I would say, by the end of today, and it will be on our website as well. So if you miss anything, you want to look at it again, just check our website. And with that, we'll just conclude then. Thank you again.
Thank you.